Good afternoon, everybody. Welcome to our edition of the BMSC Online Research Seminar Series, our fourth one of the summer, and we have a whole summer of really exciting seminars forward to. I will just give us a quick intro on who we are first, and then we will, I'm seeing that my internet connection is unstable. I hope that I'm not cutting out too much, but if I am, someone just maybe type into the chat that you can't hear me and I'll try and repeat myself. I also have the words on the screen here, so hopefully Don's connection is better than mine. Anyway, the Bamfield Marine Sciences Center is located on the west coast of Vancouver Island within the traditional territories of the Huu-ay-aht First Nation and is a not-for-profit registered Canadian charity of the University of Alberta, University of British Columbia, the University of Calgary, the University of Victoria, as well as Simon Fraser University. This BMSC seminar series is decades old and is a great opportunity to learn from experts in our community about the new discoveries being made in various fields of marine science. And it also helps support our vision to create an open, collaborative, and approachable community of scientists at BMSC and across all of marine sciences institutions. If you like what we're doing and want to see more or get involved, please visit our website at bamfieldmsc.com. So for the Q&A at the end, feel free to type your questions at any time during the talk, and Don will be able to see those at the end of the talk, so there'll be some time dedicated to answer some of those questions. To do so, just type in to the Q&A section at the bottom of the webinar of your screen. You'll see a Q&A box, and you type your question there and then hit send. So I'd like to introduce our speaker today, Dr. Don Levitan. I'm going to unmute and start video. We'll be speaking on the reproductive isolation and speciation in sea urchins. So Dr. Don Levitan obtained his PhD at the University of Delaware and conducted his dissertation on the population ecology of sea urchins in the Caribbean, or Caribbean, depending on how you pronounce that. From there, Don got his first postdoc at the University of Alberta, where he was a resident and worked at the Bamfield Marine Station, which is the old name for the Bamfield Marine Sciences Center, pre-name change branding. That's why you see BMS everywhere and those old stickers when you're on campus sometimes. Sorry for the tangents here. So this is where he started his research on the reproductive ecology of red sea urchins that he continues today. From there, he did a postdoc at the University of California, Davis, where he learned molecular techniques and more of an evolutionary perspective. And in 1994, Don started his faculty position at Florida State University and has traveled back to Bamfield almost every spring season for fieldwork and occasionally to teach a summer course at BMSC. So I think a lot of us watching can relate here. Once you come to Bamfield once, you just keep coming back almost every year if possible. So without further ado, I'm going to stop my screen share here and allow Don to start his. Welcome, Don. Thanks for being here today.
Thank you so much. I hope everyone is doing okay.
Yeah, it looks like we've got a big group on YouTube as well, so I'll manage any questions that come through there at the end if there's any YouTube questions.
Great. All right, let me share the screen and get going. All right. So what I'd like to talk to you today is about our reproductive isolation and speciation in sea urchins. And this is the culmination of about 30 years of work that I've been doing in Bamfield on the red sea urchin, Mesenchontrotis franciscanus, as it's now known. And I started off with a more ecological bent, but over time I've thought about this more from an evolutionary perspective, thinking more about how organisms and sea urchins in specific begat new species. And so the way we often define species is the ability to reproduce with one another. And so if you have two organisms and they can mate with one another, we consider them part of the same species. And if they can't mate with each other, that's one of the criteria for suggesting that they're a different species. But how does incompatibility in terms of reproduction actually evolve? It's a tricky question. The way we often think about speciation, this is a very simplified model. You can think about a single population, all these blue individuals here, belonging to one population. Then over time, some barrier splits the population in two. And this barrier we think of could be a mountain range, an ocean, a lake, islands, things like that. And once you have a barrier, it prevents genes from crossing this boundary, and the populations become isolated, and they then have their own unique evolutionary trajectory because it's independent of the other population. So one population can mutate, or both can mutate. So they change over time. And then if they change enough, when they get back together again, they can no longer mate, and we call them a new species. Further, there's an idea of reinforcement selection. And that is, if you get some changes in the population, when they come back together again, they may be able to reproduce, but if the hybrids they produce have very low fitness, they don't survive well, or they don't reproduce well, there's going to be intense selection to avoid hybrid matings, because that's all wasted reproductive effort, because offspring don't do very well. And so the secondary establishment of reproductive isolation by reinforcement is another way in which you can get speciation. So for a lot of marine organisms, this idea of reproductive isolation stems from eggs and sperm being able to mate with one another. So the mating happens in the ocean via external fertilization. So broadcast spawning, which a lot of marine organisms do, males and females release their sperm and eggs into the ocean. They meet, and they can either fertilize one another or not. And the ability to fertilize each other comes down to the proteins on the surface of sperm and eggs that recognize one another or don't recognize one another, allowing for gamete fusion. So how do you get the evolution of incompatibility between sperm and eggs that will allow for this isolation? And it's a real puzzle, because this has to happen within a population where a mutation arises in either the egg or the sperm that makes it less compatible with all the other gametes in the population. Those gametes aren't going to do very well and should be eliminated by what's called purifying selection. And so any mutation that confers a reduction in compatibility is going to be selected against. So how can you get these mutations with low compatibility actually arising in the population, increasing in frequency such that this population is now no longer compatible with another population? So how does this process occur? It's a mystery. So there's been some theory trying to explain this. And one idea is that it could be driven by polyspermy. Polyspermy is when there's so much sperm in the water that it can kill the eggs and stop development. And so under those conditions where you have so much sperm that ends up killing the egg, a mutation to that egg surface that makes it less compatible will actually be beneficial. And so here's some graphs down here that I made up. And so this is sperm concentration, and here's fertilization success, and here's a typical fertilization profile where you have low fertilization here because of sperm limitation, fertilization peaks, and fertilization decreases because of polyspermy. And so if you're a population sitting over here, you could have really low fitness. If you have a mutation to the egg surface that makes it less compatible, it shifts the distribution this way, so then you need more sperm to get fertilization, and now you have higher success compared to the old protein. Once you establish this novel egg protein in the population, it does well because it doesn't die because of polyspermy. It increases in frequency and sets up a target for a novel sperm type that matches that new egg type. And you get a generation of two compatibility groups, a paired set of sperm and eggs that match each other. And this then becomes the fodder, the variance needed to give you speciation and isolation. The first thing you need to do is get the variation so that you can have a speciation event. And so this talk will be a lot about how you generate that variation. So this idea makes two basic predictions. One is that if you have high sperm availability, that is, you live in an environment where there are lots of males releasing sperm, this should lead to the generation of multiple compatibility groups. If you don't have a lot of sperm, you just have one type. And second, across species, the new type should enhance reproductive isolation because it's a novel type that other species could recognize as being different and prevent fertilization. So those are two predictions that I'd like to talk about during the course of the seminar today. So to test these ideas, we need to know a few things. We have to know whether sperm are limiting or overabundant in the ocean. And so are sperm limiting? Is polyspermy common? What really goes on in the ocean as opposed to in a lab petri dish? Second, there's been a lot of work on these gamete recognition proteins, but most of the work just documents what those proteins are and not really how they function in terms of fertilization. And so do these proteins actually vary within a species? And does that variation actually influence patterns of fertilization? Third, is there evidence of evolution of these proteins? That's do you see evidence of compatibility groups emerging with shifts in sperm availability? And finally, the work that I'm sort of just starting now, the new direction I'm working on, is there evidence of reinforcement selection action on these proteins? In other words, are the changes that we see from these within species processes, do they have any influence on what happens in terms of fertilization across species with other sea urchin species? So I've been mostly working with the red sea urchin, Mesocyntrotus franciscanus, and this is a work I did a while ago. A nice thing about sea urchins, the reason why I work with them is you can inject them with potassium chloride, and they start to spawn within about 30 seconds. And so you can go underwater, start injecting sea urchins, and you can get a whole population of sea urchins starting to spawn. Once you get these individuals spawning, you can go underwater with an underwater vacuum cleaner, collect the eggs in the water column above each spawning female. You can map the positions of each spawning individual in the population. You can take a genetic sample so you can genotype the individuals. And then you can bring those eggs back into the laboratory, first see if they're fertilized, and then second, figure out who the mom and the dad is for all the offspring produced from these spawning events. So this is a picture from one spawning event, but I did this whole thing many, many times over a few years and generated this graph. And so each data point here is the average reproductive success for that whole population, this whole population. So I repeated that experiment many times. And so what we have here is the density of males in that spawning population and the average female success, the fraction of eggs that are fertilized. And you see the same thing that I showed with our cartoon earlier. You have low fertilization with few sperm, and that's because of sperm limitation. As the number of males increases, there's more eggs being fertilized, you reach a peak, and then you get a decrease due to polyspermy, okay, egg death due to too many sperm. And so you can see in nature a whole variety, a whole spectrum of sperm environments that might have very different selective pressures, going from sperm limitation to polyspermy. So these conditions might select for only one gamete type, and these conditions might select for multiple compatibility groups. That's the idea. So that's looking at the average success, but it's also interesting to look at the variance in success among individuals. And so this is just a representation for one of those spawning events. And I've listed all the males that spawn in that event and all the females that spawn in that event. And each cell is the pairwise reproductive success between those males and females by genotyping the offspring. And so you can sum across this way, across the column, the rows, and get the total female success. That's the fraction of eggs fertilized for those different females. And you can sum down the column to get the total male success across all those females. And looking at these patterns of fertilization, you can get an estimate of the patterns of sexual selection that you might see in this population. And sexual selection is often defined by patterns of mate variance. And what I mean by that is when we typically think about sexual selection, we think about lions or elephant seals, where you have one male winning in competition with all the other males. He sires all the offspring from all the females. And so he has really high reproductive success. All the other males have zero fertilization success, or occasionally they sneak in and get something. But it's characterized by high variance because you have a lot of winners, or a few winners and a lot of losers. Where females are always mated, and so they generally have high reproductive success. And they're not worried about getting mated. They're worried about resources, hunting, getting food, those sorts of things. And that sets up the dichotomy that we often hear in sexual selection of males competing and females choosing. And it has to do with this dichotomy in terms of the variance in reproductive success. But if you look at this data, and this is just one spawning event, you can see that the variances are way more equal between males and females. It's not what you might see with typically studied terrestrial organisms. And so now we're looking at the variance success for all those mating events. So the variance in mating success as a function of mate density. And blue is for males, pink is for females. You can see for males, you see high variance pretty much across all mate densities. Males are always competing for fertilizations, and you have some winners and some losers. For females, you have this much more complex pattern where the variance is high at low densities because of sperm limitation. Some females are fertilized, other females are not. And so you have variation. As density goes up, more and more eggs get fertilized and the variance goes down. There's no variance if all the eggs are fertilized. And so here, basically 100% of the eggs being fertilized, the variance goes down. But then the variance goes up again, but now it's driven by polyspermy rather than the sperm limitation. And so you have equal variances here and here, but it's driven by very different mechanisms. And here the selection would be for cooperation because males and females both want to enhance fertilization rates and have high compatibility. And so selection should favor just having the best compatible sperm and egg types and no variation. But here's where you have sexual conflict. And what's happening is that the males are competing for fertilizations and the females are trying to avoid fertilizations because it's killing them due to polyspermy. And these are the conditions that can lead to the rapid evolution of those gamete recognition proteins and the establishment of multiple compatibility groups that can lead to speciation. So that's the sort of the ecological backstory to what's going to go on in terms of these proteins. So now I want to introduce the proteins. And for the sperm, it's called the sperm binding protein. And these sperm are found at the head of the sperm. And they bind to these egg receptors on the surface of the egg. And they've been described for quite a long time. But as I noticed, said earlier, not necessarily the functional consequence of these proteins, just that these proteins existed. And so here is the sperm binding protein for the red sea urchin. And this is a simplified diagram showing you that there's really two places in the protein where you see variation. You see an amino acid substitution here, and it's for arginine and glycine, and here it's reciprocal glycine for arginine. And so you have the RG type, the GR type, and less frequently the GG type, and almost never the RR type. This is what's called the haplotype network, which basically shows the same thing. Each circle represents a different form of the protein. This is all within a species. And the size of the circle is how frequent it is in the population. And so this is the RG type. It's got arginine and glycine at these two places. This is the GR type. It's got glycine and arginine at those two places. This is the GG type. And like I said, the RR type is very rare. So this is the most common protein. It's close to 60% in the population. The GR is second most common. It's about 30% of the population, and the GG stays about 10%. And this is true in Banfield, but all the way down the coast from Baja to Alaska, you see these two proteins being maintained in the population. So we can look at some of the functional consequences of these two different protein types. And so this is a lab experiment where we took the eggs from one female, replicated many times, and allowed them to be fertilized by the sperm from multiple males, all pooled together. And what you can see here, see if I can move that, is that this is who wins in competition, the paternity. And the RG type beats out the heterozygous and the GR types. So the RG protein has high compatibility, high affinity for average eggs. But this high compatibility comes at a cost. Okay, this is looking at polyspermy. So sperm concentration and the amount of polyspermy you see, and all these blue dots here are high levels of polyspermy. And so you can think of that RG sperm as being aggressive sperm. It's quick to fertilize and quick to kill. And so it's high affinity. It fertilizes eggs easily, but it can also damage the eggs because it's such high affinity, it leads to polyspermy. The GR protein has lower levels of affinity to the average egg, and as a result is much less likely to cause polyspermy. And so one idea is that this GR sperm is just wimpy. It's really not very good at fertilizing, and because it's not very good at fertilizing, it can't kill the eggs. But there's another possibility, and that is that the GR protein isn't wimpy, it's just highly compatible to a rare egg type. That is, it's very compatible, but to only very few females in the population. And so to get a handle on whether that's the case or not, we have to now look at the other side of the equation, the egg receptor. So we've been talking about the sperm binding protein, and now we're going to talk about this egg receptor protein on the surface of the egg. Now, this has only been looked at a little bit more recently, and the reason is because it's a very complicated protein. It's huge. It's about 5,000 amino acids long, and so we still don't know very much about it. But I've been working on it for about 10 years now, looking for evidence that this protein varies within a species and looking at its fertilization effects of that variation. And so for most of the sites that I've looked at in this protein, there's no variation. And if there's no variation, you can't explain differences in compatibility. But in 4 of these exons with the stars, I found evidence for at least 2 different types being held in near equal frequencies. And those are the places where I thought really might be involved with compatibility because they vary and they're maintained in the population as variants. And so these are the haplotype networks for those 4 different exons. And so it's just showing you that there's basically 2 different types of TSP8. There's 2 different types of CUB1, 2 different types of TSP13, and slightly more complicated with CUB7. Those are just the names of the 4 exons where we saw the variation, and now you can see what the variation looks like. So I wanted to know what the fertilization consequences were of having these different alternate amino acids. And so we spent about 5 years doing a whole bunch of crosses in these red sea urchins in the lab to try to figure out patterns of compatibility. And I'm going to give you sort of the abbreviated answer to that question. And that is, we found evidence for compatibility groups in TSP8. And so in one of those exons, there was a strong interaction between the sperm and the egg types, giving compatibility groups. And so that RG variant, what we thought was the aggressive type, matched really well with the serine substitution, and the GR variant matched really well with the glycine substitution. And so these blue sperm and eggs have high compatibility. These pink sperm and eggs have high compatibility, and the crosses between them have low compatibility. And that's what this graph shows here. And so you have these 2 compatibility groups, and the crosses between them have much lower levels of fertilization. And so now we've got a handle on what may be explaining patterns of compatibility, and what's driving these compatibility groups is these pairwise amino acid substitutions that affect compatibility, giving you these very different groups. So what I'd like to do now is do a little bit more ecology and put this result into the context of what's gone on in the Pacific over the last few hundred years in terms of its ecology, to see if we can get a sort of understand the context of what's going on in terms of the evolution of these proteins. Does it make any sense? So one of the interesting things about the coast here is that sea otters used to be really common up and down the coast from Baja to Alaska. And the Russians came in about 1750 to about 1900 and hunted the sea otters to near extinction. And so prior to the arrival of the hunters, when sea otters were really common, the environment probably looked more like this. This is what it looks like if you have sea otters present and they're allowed to do their predation on sea urchins. They eat up all the sea urchins as well as the abaloning and lots of other things. And the urchins are at very low densities on the presence of otters. And so under those conditions, I didn't actually take this picture in 1800. It's just a kelp bed. They was probably more likely to show sperm limitation because the densities of urchins were much lower. You remove the sea otters and you get the creation of these urchin barrens. In the absence of urchin predators, the sea urchins explode in densities, and you get situations where it looks like quite a bit in Banfield now, but not so much if you go further up Vancouver Island to where the sea otters are. And you get a situation that's much more likely to cause polyspermy. We have multiple individuals per meter squared during spawning events are all spawning together. So we have this species-wide gradient of sperm availability that goes over a 200-year period. So how can we use that information to test these ideas about the evolution of gamete recognition proteins and how speciation might occur? So it turns out that these urchins can grow to be a very large old age. They show no evidence of senescence. This is work done by Tom Ebert and his colleagues. So every year they get a little bit bigger. Every year they get a little bit more fecund. And every year they show no evidence of increased likelihood of dying. They don't get, they age, but they don't get old in terms of their likelihood of living to the next year. And so these large urchins, and this is done by tagging sea urchins and looking at growth rates, that the largest urchins were conceived probably a couple hundred years ago. And so these sea urchins, if you go and dive in Banfield and you find these giant monster urchins that are, 17, 18, this one's 19 centimeters in test diameter, it's probably 200-300 years old. And so they were conceived either, before or just after the sea otters were taken out. So the other thing that's interesting about these sea urchins and these proteins is that these gamete recognition proteins only function during fertilization. They're not expressed other than on the surface of the egg and on the sperm, which means that once the sea urchin is formed, those proteins should be selectively neutral after the zygote is produced, after you get an offspring being produced. And so what that means is that if you take a tissue sample from a 300-year urchin and you genotype it, you can assay what the allele frequencies were like of the sperm and eggs when they were conceived 200 years ago. So we can look back in the past of what these proteins look like because those proteins aren't expressed and so selection can't operate on them and the urchins are still alive. And so here we're looking at the estimated age based on size. This is just the relationship between size and age for a reminder. And this is sperm binding, the protein we looked at first. This was that aggressive sperm that caused polyspermy. This was the sperm that was less likely to cause polyspermy. As you go this way towards the recent, you're going from sperm limitation to polyspermy. And so as you get the rise in urchin barrens and an increased likelihood of polyspermy, the protein that causes polyspermy decreases in frequency, and the protein that's resistant to polyspermy increases in frequency. So if you look at the recently conceived individuals, the allele frequencies are nearly the same, but a couple hundred years ago, really just had one type and then a really rare second type. This is what's going on with the egg receptor, and you see the same general pattern, but it's strongest in that same exon, which influences fertilization. And so these are the two interacting proteins. And what you can see here is that with increases in polyspermy, what it looks like is you get an escape from polyspermy by this gene increases in frequency, this one decreases in frequency, and the sperm slowly follow behind. So this matches at least the prediction of sexual conflict. You have really high compatibility types, the RG protein and that serine type. And they have high compatibility. Polyspermy starts to happening. The protein increases in frequency that avoids polyspermy, matched by a new protein that matches it. So here's sort of the summary of what's going on. You have one compatibility group under sperm limitation, increases in abundance of urchins, escape from polyspermy, a sperm protein that follows it, going from one compatibility group to two compatibility groups. And so this is a scenario that can lead to reproductive isolation and speciation. You're going from having one gamete type, sperm and egg, to having multiple compatibility groups. So what I'd like to do now is spend the last part of the talk talking about where the work is going on, where it's heading. And that's starting to think about how these dynamics within species interact with the other species of sea urchins that it co-occurs with. And so if you live in Banfield, you know there are three common sea urchin species. There's the red urchin that we just talked about, Vesicentrodus franciscanus, but there's also the purple urchin, Strongylocentrodus purpuratus, and the green urchin, Strongylocentrodus drobachiensis. And so if you go diving here, you'll see all three species, but only the red and the purple urchin go up and down the coast. The green urchin is circumpolar. And so you have a situation where you might be able to look for evidence of reinforcement selection because there are some places where the red urchin is next to the green urchin, and other places where it's not exposed to the green urchin. And that might lead to very different evolution dynamics because the patterns of competition and the risk of hybridization are going to be different in those different areas. I'm not going to talk about the purple sea urchin, at least right now, this talk, because that purple sea urchin largely can't hybridize with the other two species. But as I'll show you, in particular, the green eggs can be easily hybridized by red sperm. And you get some also in the other direction, red eggs being fertilized by green sperm. When you look at these urchins, here you're looking at the red urchin and then a few green urchins spread out in between them. When I've seen a spawning event, a natural spawning event, where there were multiple species all in the same place, when one species went, all the species went. And so it seems like the cue for spawning is phytoplankton bloom, and then a few males start to trickle out sperm, other males join in, and then the females join in. And both species, the red urchin and the green urchins, will do this simultaneously. And so there's no mechanism where they have distinct separate spawning times that might isolate the species. They all spawn in nature at the same time. And so if you do a lab experiment looking at fertilization, this is looking at fraction of eggs fertilized as a function of sperm concentration. So this green line is green eggs being fertilized by green sperm, high fertilization, increasing with sperm. This is red eggs being fertilized by red sperm, also pretty high. This is green eggs being fertilized by red sperm. This is a hybrid fertilization, and it does pretty well. And these are the red eggs being fertilized by green sperm, and it doesn't do it too often, but occasionally you get a little bit of fertilization. And so these green eggs, and to a lesser extent, the red eggs, can be fertilized by this other species, okay? But if you look at the survivorship, this is looking at green eggs fertilized by green sperm and looking at the embryo survivorship. And this is looking at green eggs fertilized by red sperm, this cross here. Basically, they almost always die out. And so these hybrids don't do very well. And these are the conditions you might expect to see, what I said earlier, reinforcement selection, where you might expect to see selection to avoid hybrid fertilization, because hybrid fertilization eventually leads to hybrid death. And so you would think females would try to produce eggs that wouldn't get fertilized by red sperm because they're just going to die. So these crosses are done where the eggs have no choice. They're put into a vial in the laboratory. They're only given the sperm from one type, and so they have no choice. But you can do experiments where you actually give the eggs a choice, and those are called conspecific sperm precedence experiments, where you have eggs and then you mix the sperm from two different males, in this case, two different species, and then you give the eggs a choice to see which sperm it prefers. And so although the green eggs can be fertilized by red sperm, the question is, in competition, are they fertilized by red sperm? And so this is the proportion of sperm from one species to the other. And so this is all red sperm. This is all green sperm. And these are mixtures of between them. And this is the fraction of green eggs fertilized by red sperm, so hybrid fertilization. This line represents the no choice line. It basically means that eggs are fertilized in proportion to the amount of sperm there is. So if the sperm is half red sperm, you'll get half hybrids. So there's no choice going on. It's just random encounters. Anything down here is conspecific sperm precedent, meaning the eggs are preferentially being fertilized by its own species. And you can see really strong evidence of conspecific sperm precedence. Even when it's being swamped by red sperm, you generally see it finding its own species of sperm and getting fertilized by that. So you see strong evidence of sperm precedence for its own species, but you also see variation. And this is what I'm really interested in, because in some cases, it seems like there isn't any choice. And so we see evidence of either strong barriers to hybrid fertilization or much weaker evidence. And so what might explain this variation? And so let's go back to these proteins, because they also play a role in hybridization. And so this, again, is that sperm binding protein with the RG and the GR protein. And this is what mostly in the population. And I haven't really talked about the GG protein, but I'm going to talk about it now because it's always around at low frequencies at about 10%. And it seems to be sitting in the middle of this haplotype network, which might suggest it's one of the older ancestral forms. So what's going on with that? And then this is that egg receptor that's compatible with this, showing you the two types. And so now we're going to look at the phylogenetic history of these different point substitutions. So Harris Lesios and Kurt Ziegler published a paper a while ago, which looked at the evolution of sperm binding for many of the echinoids. This data set has been enhanced since 2003, but they set up this pattern here. And so what I did was go through all the sequence data and just look at these 2 points in sperm binding where you have that GR point substitutions, just to look at that. And then also that TSP8, which we know almost nothing about, but we're starting to get a little bit data. And what you can see here is that that GG protein, that one that's only at 10% in the red urchin, is super common. And it's the ancestral state for all these urchins that we see in British Columbia, like the purple urchin, Pallidus, Drobacchianthus, franciscanus, all those urchins that you see diving there, they're all somewhat related. And you can see Drobacchianthus, like all these other urchins, have that GG, that protein form that's only 10% in franciscanus. And it's only in the red urchin do you see variation at these two locations. These are all fixed. For GG, you don't see variation. In this species, you do. And similarly for the egg receptor, it's fixed at that glycine substitution. And only in the red urchin do you see variation in amino acids at that location. And if you look at what explains variation in hybridization in those crosses, so looking at why in some cases you see strong barriers to hybridization, in other cases you see weak, it's related to the frequencies or the identity of the male having different sperm binding proteins, and these are red eggs being fertilized by green sperm and showing the identity of the egg receptor in those red urchins. And in both cases, the ancestral protein, the GG protein, is the one that leads to hybridization. Whereas the novel derived proteins, those RGGR combinations, are not causing hybridization, as well as that SS. And so what that means is that if you've got a type that's similar to Drobochiensis, it can hybridize it. If you have a type that's different than Drobochiensis, it's recognized as being different and prevents hybridization. And so the novel proteins are recognized by the other species as being different and are prevented from hybridizing those eggs. But if they have the same protein as the green urchin, they fertilize it fine, causing hybridization. So this notion of conspecific sperm precedence depends on eggs having a choice. And so do eggs actually really have a choice? We know that in the laboratory, you can force eggs and sperm together in different combinations. You can mix sperm from different males together and put them on eggs. But in the ocean, do the sperm actually see the sperm from multiple males or multiple species at that same instant when fertilization occurs? And so this is some field experiments I want to talk about before we finish up, to try to look at what the pattern of competition is actually like in the ocean. And so what I did as an experiment is I set up a bunch of males underwater, got them to spawn. And here you can see males spawning. And look at all the sperm being released. If that sperm hits an egg, it's going to be swamped by that one male. There's no competition. That male is going to win. So how do I see whether eggs surrounding all these spawning males were actually seeing competition or not? And the way I did this is I wanted to look at the environment in which fertilization takes place by collecting tiny water droplets during these spawning events that are about the size of an egg. So about 10 to 100 microns of water, which is about the size of the population of water that's going to interact with a single egg. So this is the cartoon of what the egg to that water sample look like, showing you that the sperm are likely to be the population of sperm interacting with that egg. I would collect that water droplet into a syringe. The syringe had a bunch of unfertilized eggs. The sperm or the water would come in here. Any sperm in that water would fertilize these eggs. I would then genotype these eggs and see if I could detect the sperm from either one male or more than one male, to see whether in this population of sperm, whether we could detect the sperm from more than one male surrounding individual eggs. And so I did this over a few years where we had a bunch of spawning males in different configurations, and we would collect water droplets in various places around those things, bring them back to laboratory, and then ask in these water droplets, can you detect the sperm from just one male, like the closest, or do you see sperm from all these different males, all in that same little water droplet, meaning that that egg would have the availability of choosing among those different males. If the only sperm you see from this water droplet comes from this male, those eggs have no choice. So here's a little video showing what that experiment looked like. So here you can see male urchins after injection. Within about 30 seconds, they start spawning. And you can see these clumps of sperm. If a clump of sperm hits an egg, there's no competition. That male's going to win. But as you look further, you can see that as the turbulence goes back and forth, the sperm just flows outward and starts to mix and starts to dilute. And as it dilutes and mixes, potentially you see sperm from multiple males. And so here I am taking a tiny little syringe, taking a tiny little droplet of seawater. This is a syringe with a screw. So I just turn the screw a half turn. It sucks in at like 10 microns worth of water, put it back, grab another syringe, take another sample, bring those eggs back to look at paternity, to see whether you can detect the sperm from multiple males within those tiny drops. This is the whole paper. I just want to show you one figure. This is the paternity share, what you find in a particular syringe. And so this is how successful the most successful male was in a syringe. And so if it's way over here, that means that that male swamped that syringe. And occasionally you do see that. But more often than not, you see multiple males getting some paternity, even in those tiny droplets of seawater. And that likelihood increases as you increase the crowding, either the nearest neighbor distance or the density of those urchins. So this suggests that in most cases, when you have multiple males spawning, the sperm is mixed well enough so that individual eggs have a choice. And if they have a choice, then they can exhibit that choice in terms of choosing their own species rather than a different species of sperm. And so that means that that process of conspecific sperm precedence is likely to be really important in determining whether hybrids get produced or not. The last thing I want to talk about, and this is the work that I really need to do next, and so I'm hoping to come to Banfield next year to follow up on this work. I'd like to start to do some experiments up and down the coast, but what I already do have is data on allele frequencies of these proteins up and down the coast. And the idea is that in Banfield, because you see the green and the red urchin together, you should be selection against hybridization. And the proteins that resist hybridization should be really common. Well, down here, in this case, Santa Barbara, there are no green urchins. And so those urchins shouldn't have to worry about hybridization. Selection shouldn't favor resistance against hybridization. And the proteins that allow for hybridization can run rampant because they're not going to have a detrimental effect. And that's what we're starting to see. And so this is looking at the egg receptor. And this is what we see in Banfield. This is the protein that resists hybridization being really frequent in Banfield, where hybridization is likely. But in Santa Barbara, basically, it's very, very low frequencies. And so this is what's called reproductive carrier displacement, where the allele frequencies are changing potentially due to the risk of polyspermy. Due to the risk of hybridization. The sperm protein is much more subtle, but you see the same sort of pattern. That GG type, which was really rare in Banfield, is slightly more common in areas like Santa Barbara. And that protein that doesn't cause, resist hybridization, decreases in frequency as you go into allopatric populations further south where the green urchin isn't. And so this suggests that the mechanisms that promote the diversification of these proteins also have ramifications for isolation among species. And so where this work is heading is really trying to get at, how you get speciation by working at these proteins where we're sort of in the process of working itself out. So in conclusion, high density and the risk of polyspermy can generate diversifying selection and the creation of compatibility groups, which is, I think, sort of the hardest step in trying to understand speciation. Once you have the variation, it's easy to imagine how these sort out into different species, but how do you get that variation in the first place? Novel compatibility proteins are recognized by other species and hindered hybrid fertilization. So this within species process can have ramifications across species and lead to isolation with other species. And so sexual conflict can promote assortative fertilization within species and reproductive isolation among species. And so a large cast of folks have helped out with this work I've been doing this for about 30 years now. And so people helped out in Banfield, people helped out in Tallahassee doing the molecular work, people have helped out doing the theory, and I couldn't have done it without all their help, and the work has been supported by NSF. So with that, I will end and take your questions.
Thank you very much, Don. do you have the Q&A at the bottom there? Do you see there's a 4 beside the Q&A section? If you click on those, you'll be able to sort of pick and choose which ones you want to answer.
All right, I will start off with the first one by Kylie. Hi, Kylie. So the question is whether in that mating success table, could you predict success based on nearest neighbor distance? In other words, are the closest males the ones that are always fertilizing females? And you can explain the variation based on distance, which is a great question. And the answer is some amount of variance can be explained by nearest neighbor distance. Some amount of variance is explained by the distance, by the flow conditions, which way the current is going. I didn't have time to talk about this, but I got a whole paper talking about the fact that a lot of the variance is explained by these gamete recognition proteins. So proteins that are highly compatible do much better than expected at distance, and proteins that are less compatible do far worse than predicted based on distance. So it's a combination of gamete recognition proteins, water flow, and nearest neighbor distance. Here's the second question by Kylie. We are hearing that urchin roe is declining in quality by urchin harvesters. I'm guessing it's due to collapsing kelp populations and overpopulation. Have you seen any evidence of this? And do you think you have any thoughts on what this might be due to the populations and evolution? I've certainly seen a lot of talks and heard a lot of talks, seen a lot of data about how urchin barons, if they're really starved, the gonads become really wafer thin and they don't reproduce very much. My experience in Banfield is that, this all happens on a very small spatial scale. So if you go diving at a typical site in Banfield, you'll see in shallow water a bunch of urchins all munching on algae right near the intertidal. And those are pretty well fed and they've got nice, thick, healthy gonads. But as you go down the slope, even 10 meters or so, you'll get to a place without kelp and those urchins will be less fecund. And so if the urchin barrens get so severe that males aren't really producing much sperm, instead of polyspermy, you'll go back to sperm limitation. But it won't really be sperm limitation because you're not getting any eggs either. You're just getting nothing. So I do think that those sorts of things would have ramifications in terms of the evolution of these proteins. But a lot of these things are at a very small micro scales because you can just go up and down going from shallow to deep and see very different patterns of food availability. Okay, Stacy has a question. Do you know of anyone looking at gamete gene frequencies of red urchins in areas where otters have been reintroduced and urchin populations are likely returning to historic levels, such as BC, Central Coast? I have been collecting specimens up the coast, Haida Gwaii, and down Washington, Oregon, and in California. Like I told you, I report on some data from Santa Barbara. And in various places, you can get different densities of urchins driven by sea otters and perhaps other things. The problem with trying to detect local adaptation in things like sea urchins is that the larvae go great distances before settling. And so if you just look at neutral microsatellite markers and you try to look at patterns of population structure in things like the red sea urchin, you see very little evidence of population substructure because there's so much gene flow going up and down the island. And that makes it really hard to show for evidence of local adaptation, which is why I went to this really 200-year temporal thing, because it happened across the whole species. And it was a huge selective hammer that you can start to get a grip on. I do think that, we're starting to see like those differences in allele frequencies in that egg receptor in Santa Barbara versus Banfield would suggest that, perhaps if the selection agent is strong enough, like the risk of hybridization, you might be able to detect some patterns. So the short answer is that I'm starting to look at that question, but local adaptation will be confounded by high levels of gene flow. I got a question of Joanne Lessard. In sea otter areas, theory is that urchins are in cracks and crevices and therefore aggregations. Wouldn't polyspermy be a problem as well? Great question. So purple urchins are characterized by living in cracks and crevices. When you find them, they're usually highly aggregated. Intertidally, they're aggregated. And I suspect in sea otter areas, if you had a crack or a crevice, you might get 20 or 30 urchins just packed in that crack and crevice. The purple sea urchin has gamete recognition proteins, which suggest that it's constantly under a threat of polyspermy. And so it has many more compatibility groups than the red urchin, high levels of diversity. And I've shown that they are often under conditions that might lead to polyspermy, but because their gamete recognition proteins are so polymorphic, they're more resistant to polyspermy because they have so many different proteins. And so I think it's a great question. I think the red urchin, by its nature, the way it grazes, is more likely to be sparser in sea otters, but the purple urchin, which aggregates, I think will be subject to polyspermy, even in the presence of sea otters. Good question. Would it be possible to make the relation between protein frequencies and age to make inferences about past urchin population densities? I work in Quebec. Kathleen McGregor. I think it would, but you worry about circular, reasoning. I'm using inferences about what I think is going on in terms of urchin densities to make, hypotheses about how the proteins are evolving. I wouldn't want to go backwards and do the same thing. That would be circular reasoning. But, once we really understand these proteins really well, conceivably, we can go to other areas and perhaps make some inferences. So I think that would be really fun to do. Ross Whippo says, purple urchins don't often hybridize with greens. Can you speak as to why this might be the case? My guess is because they have more divergent, highly variable gamete recognition proteins. And I think because they're subject to polyspermy, they're very, their eggs are set up to be very choosy, and they're likely only fertilized by their own species. So I just got a grant, found out yesterday, to work on this problem. And so I want to answer that question. And so the work that I'm doing now between the greens and the reds, I want to do for all these species to try to figure out which proteins might explain variation. So ask me in 5 years. So here's the question from Daniel. Very cool, blah, blah, blah. Is it known if the egg receptor proteins that make the egg resistant polyspermy degrades over time? There is no evidence that it degrades over time. That doesn't mean that it doesn't degrade over time. I think the lifespan of an egg is pretty short. And so the egg is released. And if you keep eggs around for about 5 or 6 hours, they just degrade because the bacteria eat them up. If the egg is fertilized, it becomes less permeable, right? It only has to be permeable before fertilization to allow the sperm in. Once the egg is fertilized, the outside hardens up, becomes resistant to degradation. So I don't know if there's really much time to allow for this degradation. But I do think it's a fascinating question to see how compatibility might change with time. There's been a lot of speculation about the likelihood of self-fertilization or not in these species because they can't self, but whether those processes change over time as the egg becomes more desperate. Is it more available for different types of sperm?
And Don, this will be the last question here since we're at 2:30. So we have this one more question from Dan. And
right, how does this speciation differ from drift in the population from one steady state to another? So I think a lot of characters can drift, but for gamete recognition proteins to drift, it means that selection has to be unimportant. And a lot of what my work has shown and others' work has shown is that eggs are constantly facing the devil of the deep blue sea. Either they're faced with sperm limitation, too little sperm, or polyspermy, too much sperm. And there's very few conditions in which the eggs are like in that perfect peak where they have just enough sperm where all the eggs are fertilized. And I think because of that, I don't think it's likely that these gamete recognition proteins are likely to drift. So lots of traits might drift, but I think the evolution of reproductive compatibility, I think, has to be driven by selection.
great. Thank you so much, Don. I'm just going to share my screen here as well. I wish we had a function for us to give you a round of applause here, but we can only assume that it's a deafening roar of clapping and applause. So yeah, absolutely. Thank you so much for joining us today and enlightening us on your research. And we're really excited about seeing what's coming up next. Congratulations on your grant that you found out about yesterday.
Yep. Thank you so much.
Thank you. Okay, well, I'm just going to share my screen here to enlighten folks about the next week's seminar.
Bye.
See ya. So there's your thank you slide. And okay, so next week we're going to be speaking to Julia Baum. Dr. Julia Baum is going to be talking to us about in hot water. So we're really excited for that. You can check out Julia's research page. You'll be able to find her on Google instantly if you put her name in to sort of get prepped for the talk if you like. And then tune in at the same time, 1:30 p.m. Pacific Daylight Time, June 23rd. Thank you everyone for joining us today, and I hope you have a great week. Okay, bye-bye.